Hi guys, today we're going to talk about carbohydrates. You should have your graphic organizer ready and fill it in as we go along. Carbohydrates are pretty easy to recognize because they always have a reduced formula of CH2O. So that means just like reducing a fraction, if you reduce the formula down of C6H12O6, it will reduce into CH2O. So the hydrogen and the oxygen are in the same ratio as water. The monomer of a carbohydrate is known as a monosaccharide. The dimer is called a disaccharide, and a polymer is called a polysaccharide. And on the next few slides here, we'll talk about examples of each of these. In living organisms, there's two main functions for carbohydrates. The first is something that is not a function in your body, but is important in many living things, and that is structure. In some organisms, especially those that don't have skeletal systems like us, they use carbohydrates for structure. Insects, for example, their shell is made of a polysaccharide called chitin. Plants don't have bones either, and a lot of their structure comes from their cell walls. The molecular component of the cell walls that gives them that firm structure is called cellulose. Again, these are both examples of a polysaccharide, really long chains of carbohydrates that provide structure for these organisms. The second major function is that they provide energy. This is an immediate energy in the form of a monosaccharide, so you eat sugar, you get energy right away, or a stored energy if it's in the form of a polysaccharide. We already talked about cellulose being a polysaccharide in plants, but it's important that you also know the polysaccharide that's found in animals, which we call glycogen. So in your body, if your liver stores sugar long term and converts it into a polysaccharide, it would be in the form of glycogen. Now, looking a little bit more closely at some examples of these different molecules, monosaccharides, disaccharides, and polysaccharides, there are different examples that we want you to be aware of. Now, all the monosaccharides are going to have this same structure that you see here, and they are going to have, when I look at that, I see a hexagonal ring. Their general formula is always C6H12O6, but as we've already discussed the term isomer, different monosaccharides will be isomers of each other. So they have the same formula, but different structures. The most common examples, and the ones that I want you to know, examples of monosaccharides are glucose, fructose, and galactose. These are single unit sugars, and they taste very, very sweet. Looking at the disaccharides here, we see two rings of two monosaccharides linked together. Right here, draw an arrow for you, is where that condensation reaction would have taken place, and you'll model this in your lab. As they come together, one water would come out and form. So you have a monosaccharide plus a monosaccharide yielding a water and a disaccharide. Now the molecular formula for a disaccharide is not perfectly multiplying this C6H12O6 by 2 because of that loss of water. So if you look at the formula, we had to subtract out an H2O. You can use this formula and scale this up for any polysaccharide that you want. I could tell you, for example, that there are 100 monosaccharides linked together to make the polysaccharide of cellulose, and you could calculate C6H12O6 times 100 and then subtract out the 99 waters that would have formed. The examples of disaccharides for you that you need to know are sucrose, maltose, and lactose. Again, some words that should be somewhat familiar to you. Sucrose is table sugar, lactose is the sugar in milk. These are still things that would taste sweet to your taste buds. And then finally, the polysaccharides we see here in D, linking together multiple, 3 or more of the monosaccharide monomer units. We already talked about the fact that there are many different polysaccharides that you should be aware of, glycogen and cellulose and chitin from the previous slide just being a few. So if I were to give you in class tomorrow 3 different solutions and tell you that one solution was a monosaccharide, one solution was a disaccharide, and one solution was a polysaccharide, you should be able to take the knowledge that you have from this short lecture and determine which cup or which solution was the mono, the di, and the polysaccharide. Because the monosaccharide is so small, it would be the sweetest on your taste buds. The polysaccharide is large and would require enzymes to react to do hydrolysis reactions and break down that large molecule before the sweetness could be tasted. If you want to test this out, get a saltine cracker from your pantry at home and chew it up. Keep it in your mouth for a few minutes and let the enzymes in your mouth work on that polysaccharide. And it'll start to go from a starchy cracker bread taste to a more sweet mono or disaccharide taste on your taste buds after a few minutes. In the lab, you will need to be able to test for both monosaccharides and polysaccharides. The tests are pretty simple. For the monosaccharide test, you'll use an indicator called Benedict's. You can see here in the picture, Benedict starts off as a blue color. You want to add equal parts of the solution and the Benedict's together, so probably start off with something about 20 drops of each. And then you, for this test, have to place your test tube into a hot water bath to heat it for a few minutes. The hot water bath does need to be pretty warm, but it does not need to be boiling. If a sugar is present, the Benedict solution and the sugar will turn into one of these bright colors here. That bright red, orange, or green can all be an indicator for the presence of sugar. If there is no sugar present, it will remain the same. For polysaccharides, the test is even simpler. We use iodine. This is the same iodine that you have probably seen in a doctor's office before. For this test, you just need to add equal parts of the iodine in the test solution. Because this one does not get heated, you can simply put it onto a spot plate. So you can use just a few drops of each. If the iodine turns to a dark purple, almost a black color, you know that starch is present. So in this case, the picture is backwards. Here's what the iodine originally looks like. It's a yellowish color. If it changes to that dark color, you know that starch is present. One suggestion that I would make for you with both of these tests is to run a control next to your known substance. What I mean by that is to add equal parts of Benedict's and water to a second test tube, or to add equal parts of iodine and water to your spot plate so that you can definitely see what the negative test looks like. It'll help you to understand and interpret the positive results if you can see what a negative test looks like right next to it.